Unit 7, which is microevolution and macroevolution. So, macro and microevolution have around 13 to 20% of questions on the AP exam. So, for microevolution, this is going to be natural selection, evolutionary forces, sexual selection, Hardy Weinberg equation, inbreeding coefficient equation. Then for macroevolution, this is going to be history of life, geological timescales, common ancestry, and cladistics. So for microevolution, we're going to be starting with natural selection. So with natural selection, this is when traits that bring higher, greater reproductive successes will increase frequency over generations. So natural selection was originally proposed by Alfred Russel Wallace, who caused Charles Darwin to release his book on the origin of species in 1859. So natural selection can only occur if one, a population has variation. If a trait is advantageous and helps the individual survive and reproduce, the genetic variation is more likely to be passed to the next generation. Secondly, is a force causing selection to occur in the first place. So, a force will cause environmental pressures to favor certain traits that are passed on to offspring. Third would be heritable traits. There has to be heritable traits that must increase the organism's probability of surviving and reproducing. Secondly, to be passed down, phenotype traits are not inheritable, only genotype traits. All three of these must be true and available for natural selection to occur. So, one of Darwin's inspirations is Thomas Malthus's Principles of Geology. Malthus said that natural processes have always been occurring. This definition has also been coined under the term known as uniformitarianism. So, processes such as photosynthesis or erosion have always been occurring. It's also important to remember this principle because you can cross apply it to other aspects of science. So, beyond natural selection, there are three types of selection we must also remember. So, first would be artificial selection, is when humans select who to breed. So, for example, in recent decades, a lot of dog breeds have been artificially made through dog breeders. Secondly, would be sexual selection. This is when mates are selecting who to reproduce with. Oftentimes, we see females of certain species choose male partners that are more than likely to be dominant enough to produce viable offspring. So, certain behaviors or traits will cause and attract females to reproduce with these males. Third would be convergent evolution. This is when selection leads to unrelated species having the same or similar trait. So, for example, in butterflies, some unrelated species of butterflies have the exact same trait, which could range from having the same wing pattern, size, or color. And this occurs because of the similar conditions that both species have been put in, and the same selection force has been similar, causing them to have the same trait, which would be known as convergent evolution.
All right, so next we have evolutionary forces. There is four different evolutionary forces. Can be natural selection, mutation, drift, or migration. And so natural selections is a non-random process. Mutation is random, drift is also random, and then migration can happen both randomly and non-random. We have natural selection, which is basically survival of the fittest trait. This refers to traits or characteristics that confer a reproductive advantage, leading to greater success in passing on genes to future generations. So, certain alleles lead to better success in certain environments, which indicates that specific allele are more advantageous in particular environmental conditions. which increases the likelihood of survival and reproduction for individuals carrying those alleles. So, natural selection can affect traits influenced by a single gene or polygenic traits as well. So, when discussing evolution, we use a word called fitness. And what this refers to is it describes the measure of an organism's reproductive success relative to others in a given environment, influenced by its traits and abilities to survive, reproduce, and pass on those genes to offspring ultimately. All right, so there's different types of selection patterns. In disruptive selection, both extreme phenotypes are favored, indicating that individuals at the far ends of the phenotypic range have higher fitness compared to those with intermediate traits. This process drives the divergence of traits within the population, leading to the prevalence of extreme phenotypes. However, in directional selection, only one extreme phenotype is selected for. And this highlights its superior fitness over alternative traits and results in a shift towards that extreme within the population. And then lastly, we have stabilizing selection, which favors intermediate phenotypes, indicating that individuals with traits closer to the mean possess higher fitness relative to those at the extremes. This stabilizes the distribution of traits within the population, maintaining genetic diversity while reducing the prevalence of extreme phenotypes. We have frequency dependent selection, which is where rare traits eventually become common, and this because rare traits are selected for over common traits. And the cause of this is typically because predators tend to focus on more common traits. So, an example of this we can see in this background right here. So, we have big orange butterflies and smaller yellow butterflies. So, let's say the predator, which is this crow right here, only sees the big orange butterflies and it preys on those. These orange butterflies will then reduce in population, and the rare yellow butterfly phenotype will increase, making them the common species and traits in the population. Okay, moving on to the next evolutionary force, we have mutation, which occurs very, very, very slowly, and it is the only evolutionary force that can create new traits. Now, because of this, mutation will not make a trait common by its own. So, selection or drift can help make a mutant trait more common in a population. Drift, which is random changes in allele frequencies. They are more noticeable in smaller populations due to the increased influence of chance events on allele frequencies. However, larger populations are less affected by drift since random fluctuations are spread out across more individuals. So then we have the founder effect, which happens when a new population starts with only a few individuals, reducing genetic diversity compared to the original population. And then we have bottleneck, which is a bottleneck of event occurs when a population undergoes a significant drop in size, which results in a loss of genetic diversity and potential long term effects on the population's ability to adapt to its environment. So, lastly, we have migration, which involves the movement of individuals from one population to another, resulting in the transfer of alleles from one gene pool to another. So, this process can introduce new genetic variations into a population, or it can also alter existing allele frequencies, which ultimately influences the population's genetic diversity. So, by studying patterns of allele flow between populations, biologists can gain insight on how migration shapes the genetic structure of populations over time and its implications for adaptation and speciation. Right. Next, we have sexual selection. So, sexual selection is a type of natural selection based on acquiring mates. There is two types. There is intersexual selection and intrasexual selection. Intersexual selection is when one gender chooses mates from the opposite gender, usually with females as the choosing sex. And then intrasexual selection is competition among individuals of the same gender for mates, often with males as the competing sex. So next we have sexual dimorphism, which is a result of sexual selection. So sexual selection leads to physical differences between sexes, driven by reproductive needs and mate selection. These differences often result in elaborate traits in the sex facing less competition for mates. For instance, think of the peacock's tail or the vibrant plumage of the male birds. These traits evolve through mate preferences or competition, shaping the disparities observed between the males and females in many species. Next, we have the role of gender in sexual selection. So, in reproduction, males typically invest fewer resources and possess unlimited reproductive potential, making them more competitive in mating scenarios. However, females tend to allocate more resources towards their reproduction and they have limited reproductive potential. So, this makes them more selective. And then we have the good genes hypothesis. It suggests that females select mates based on traits that indicate male fitness, driven by the desire to secure advantageous genes for their children or offspring. However, it's important to note that females are not objectively assessing males for their genetic quality. Instead, their preferences are shaped by natural selection. So, where traits selected by females confer fitness advantages and ultimately contribute to the reproductive success of both sexes. All right, so now we have mating systems. So, the first type is promiscuity. This is when a member of one sex mates with an individual from the opposite sex. So, female choice and male competition will still be present during promiscuity. And then we have monogamy. So, monogamy is when one male pairs with one female. It's typically prevalent among birds, and this arrangement is often linked with the shared responsibility of both parents in caring for their offspring. So in monogamous like bird species, both the male and female participate in raising the young, which is crucial for the survival of the offspring. This long term commitment to a single partner is also known as long term monogamy. So, there is long term monogamy and serial monogamy. So, long term monogamy refers to a mating systems where individuals form a single pair bond and remain together over an extended period, often for multiple breeding seasons or even for their whole entire life. But then, in contrast, serial monogamy involves individuals forming new pair bonds for each breeding system or each breeding season or reproductive cycle, with the possibility of switching partners between seasons. So, as males become increasingly scarce, there is typically a shift in traditional gender roles. So long-term monogamous males tend to prefer partners with youthful neotonic traits because younger females offer more reproductive cycles, and serial monogamous males lean toward selecting mates with mature paramorphic traits. This preference may be influenced by the fact that older females with accumulated experience in raising offspring offer valuable parenting skills. So therefore, as the scarcity of males alters the dynamic of mate selection, both sexes adapt in their own ways to optimize the reproductive success. And then lastly, for sexual selection, we have extrapair copulation. So the concept of extrapair copulation, or EPC, illustrates how individuals may employ alternative mating strategies to maximize their reproductive success. So one male gains an extra offspring through EPCs with females other than his primary mate, enhancing his overall reproductive success. At the same time, one female benefits by engaging in EPCs with males possessing superior genes, thereby ensuring better genetic quality for her offspring. However, one male remaining faithful to his monogamous relationship receives no reproductive benefits from EPCs, which highlights the differential outcomes of the mating strategies within a population.
So, in order to solve for Hardy-Weinberg, there are five assumptions that must be true in order to calculate and solve for Hardy-Weinberg equilibrium. So, first would be a large slash infinite population. Oftentimes, we must be assuming infinite population to that must be precisely infinite because a quantifiable population can be limiting. Secondly, would be random mating, which means that all organisms in a certain species have an equal chance at mating compared to the next person in the same species. Third would be no mutations occurring in the species. Fourth would be no migration or gene flow, meaning that they've been living in the same spot and they're not moving or no one is coming to them as well. And then fifth would be no selection force causing them to change as well. We cannot solve for equilibrium if one assumption is false. A population that is in Hardy-Weinberg equilibrium is not evolving. This means that the allele and genotype frequencies do not change as generations pass. So now on to the Hardy-Weinberg equation. The prerequisite to using the Hardy-Weinberg principle is that the allele frequencies must add up to 1. P, which is the homozygous dominant allele, plus Q, which is the homozygous recessive allele, must be equal to 1 to represent the entire population. Because there are only two possibilities for alleles and they have to add up to 100%. Now, if we know the allele frequencies, we can predict the genotype frequencies. And on the actual equation itself with the Hardy-Weinberg principle, as seen in the picture right here, is that the frequency of homozygous dominant genotype would be our p squared, whereas the frequency of our homozygous recessive genotype is our q squared. And then 2 times p of q would be the frequency of the heterozygous genotype. And all of this must be equal to 1 in order to represent the entire population. Now, a key tip Mr. Cahill taught us was to solve for Hardy-Weinberg equilibrium by finding the q squared, or the frequency of homozygous recessive genotype, first, because it makes it much easier to solve. Though it may seem like basic algebra solving for variables, never use Hardy-Weinberg to find the p and q because you cannot assume equilibrium even if all five assumptions are true. So now on to the inbreeding coefficient. So there are two types of breeding, inbreeding and outbreeding. What is inbreeding? So inbreeding is the breeding of two closely related individuals, which is bad because similar genotypes can cause offspring to be affected by recessive traits. Oftentimes, this occurs because of limited population size. And then this can also lead to inbreeding depression, which is when the population becomes less healthy as more recessive traits are expressed. Then what is outbreeding? Outbreeding is the breeding of two distantly related individuals, which is good because it increases genetic diversity and could potentially bring elite alleles into the breeding population or gene pool. So now when we solve the equation for inbreeding coefficient, when we solve for it, is going to be f is equal to 1 minus the observed frequency, or the heterozygous frequency which is observed, over 2 times p times q, which is the expected frequency or expected heterozygous count as well. So now there are four things we must remember for the inbreeding coefficient. If f is equal to 0, that means that there's no inbreeding at all. If F is less than zero point 0.1, this means that there's an acceptable level of inbreeding that's happened throughout the species. If F is greater than zero point 0.1, that means there have been high levels of inbreeding in the species. Then, if F is less than 0, that means there has been excess outbreeding, which is entirely too good or entirely too bad, but it's just right.
Macroevolution is about small changes within a species. For example, it includes changes like some birds having slightly longer beaks than others. These changes happen over a short time and within one species. Macroevolution refers to bigger changes that can lead to new species or major transformations, like reptiles evolving into birds. This kind of evolution takes a much longer time. The History of Life, the Miller-Urey experiment, a landmark scientific study, recreated early Earth's chemical conditions in a lab setting, converted simple inorganic molecules into vital organic compounds. Ingredients included NH3, CH4, H2, and water, resulted in the formation of amino acids, sugars, and lipids. These chemical reactions in the early oceans are dubbed as the primordial soup. Jean's first hypothesis states that the origins of life are believed to begin with self-replicating RNA molecules. These RNA molecules would have been subject to evolutionary pressures, leading to the development of more complex RNA organisms over time. Eventually, RNA organisms would incorporate other macromolecules into their systems. DNA emerged as the primary carrier of genetic information, supplanting RNA's role in this regard. RNA exhibits a remarkable versatility in biological functions. It can store genetic information as seen in viruses. RNA can also store energy within its molecular structure. In the form of messenger RNA, RNA serves as a conduit for transmitting genetic information from DNA to protein synthesis machinery. MicroRNA molecules regulate gene expression by targeting specific messenger RNAs for degradation or inhibiting their translation. Ribosomal RNA catalyzes key reactions in the process of protein synthesis, highlighting RNA's catalytic capabilities. Metabolism first hypothesis states that the initial step in the origins of life involved the formation of self-sustaining metabolic reactions. These reactions relied on energy and essential components obtained from the environment, such as hydrothermal vents or other geochemical sources. Over time, additional components were incorporated, leading to the development of more intricate chemical networks. Many metabolic reactions are cyclic in nature, allowing for a continuous flow of energy and resources. Once established, these self-sustaining reactions would persist and evolve under the influence of evolutionary forces, gradually becoming more complex and diverse. The extraterrestrial hypothesis suggests that life originated from sources beyond Earth. In its more cookier interpretation, the hypothesis proposes that the fully formed extraterrestrial life somehow arrived on a planet like Earth, capable of supporting its own unique form of life. In a less cookier version, the hypothesis suggests that the important components necessary for life's emergence, such as organic molecules or even primitive life forms themselves, were delivered to Earth from space. While less conventional, the extraterrestrial hypothesis offers an alternative perspective on the origins of life, sparking debate and further exploration into the possibility of life's cosmic origins. Evidence of macroevolution: Anatomy and embryology: Similar structures and developmental processes in different species suggest common ancestry. Molecular biology: Genetic similarities between species reflect shared ancestry and evolutionary changes over time. Biogeography: Distribution of patterns of species indicate evolutionary divergence and speciation events. Fossil record: Fossilized remains provide evidence of evolutionary transitions and the diversification of life. Geological timescales: Understanding the basics, the eons, which include the Hadean eon, which was also involved with the formation of the Earth, the Archean eon, which was crust solidification, the Proterozoic eon, which is supercontinents, and Phanerozoic eon, which was multicellular life explosion, to present day. The geological timescale in the Precambrian era included the Hadean eon, in which there was no life on Earth, the Archean eon, where prokaryotes appear, and the Proterozoic eon, where eukaryotes appear. For this unit, we only have to memorize three dates, which are the Cenozoic era, the Mesozoic era, and the Paleozoic era, in which the Cenozoic era extends to present day from 65.5 million years ago, the Mesozoic era extends from 65.5 million years ago to 245 million years ago, and the Paleozoic era extends from 245 to 570 million years ago. Starting off with the Paleozoic era, the Cambrian period witnessed the Cambrian explosion, a rapid diversification of multicellular eukaryotes. Arthropods, notably trilobites, emerged as the most dominant animal group. Significantly, some organisms ventured onto land, marking an early phase in the colonization of terrestrial habitats. The Ordovician experienced the Ordovician radiation, another significant diversification event comparable in scale to the Cambrian explosion, marked the appearance of jawless fishes, representing a crucial step in the evolution of vertebrates. The Silurian witnessed the appearance of bony fish, marking a key advancement in vertebrate evolution. First, vascular plants emerged, contributing to the greening of terrestrial landscapes. It saw the appearance of the first fully terrestrial animals, marking a significant transition in the colonization of land. Notably, Pneumodesmus, an early millipede-like arthropod, represents one of the first fully known terrestrial animals. The Devonian, which was also referred to as the Age of Fishes, characterized by the extensive reef systems contributing to the diversification of marine life, saw the emergence of early forests, composed primarily of primitive plants like ferns and mosses. Climate variation during this period may have played a role in shaping evolutionary trends, including the transition of fish to terrestrial tetrapods. The Carboniferous, which was divided into the Mississippian and the Pennsylvanian, was when the amphibians undergo diversification. The first amniotes, the egg-laying vertebrates, emerged. The Lycopsid forests proliferate. Giant terrestrial arthropods like Arthropleura inhabit the landscape. The Permian was when gymnosperms, the first seed-producing plants, appear, paving the way for the evolution of modern conifers and cycads. Gymnosperms gradually replaced lycopsids and fern trees, reshaping terrestrial ecosystems. Synapsids, a group that includes early mammal ancestors, become the most dominant land vertebrates. Mammals, which are descendants of synapsids, begin to emerge. The first dinosaurs appear, representing a significant evolutionary development in reptile diversity. The period concludes with the Permian-Triassic extinction event, the most severe extinction event in Earth's history, resulting in the loss of approximately 95% of all species. A little things to know about the Permian-Triassic extinction was that it occurred 252 million years ago, resulted in the loss of about 95% marine species and 70% terrestrial vertebrates. It was likely caused by massive volcanic eruptions and subsequent climate change. A few mnemonics to memorize the Paleozoic era are: "Can otter sing during mating periods, purring majestically?" or "Come over some day, maybe play poker?" or Cahill often says, "Dinosaurs make parties perfect." Moving on to the Mesozoic era. Starting with the Mesozoic era, we have the Triassic, in which the synapsid crocs dinosaurs vie for dominance after the Permian-Triassic extinction event. Dinosaurs emerged victorious, establishing their dominance over terrestrial ecosystems. Dinosaurs started diversifying into various forms, paving the way for their eventual dominance during the Mesozoic era. Mammals make their first appearance, marking the beginning of their evolutionary journey alongside the dinosaurs. After the Triassic, when the Jurassic comes, we see that gymnosperms, including conifers and cycads, dominate terrestrial flora. First angiosperms, or flowering plants, possibly make their appearance, though not yet dominant. First birds emerge, representing a remarkable evolutionary development from theropod dinosaurs. Pangaea, the supercontinent, begins to split into separate land masses. Dinosaurs undergo a phase of gigantism, with some species reaching enormous sizes. The Cretaceous was when the angiosperms or flowering plants became increasingly prevalent, reshaping terrestrial ecosystems. Certain dinosaur groups faced extinction, leading to shifts in biodiversity. Stegosaurids, characterized by their distinctive plates and spikes, roamed the land. New dinosaur groups emerged, including ceratopsids, known for their elaborate frills and horns. The Cretaceous-Paleogene extinction event occurs, resulting in the demise of approximately 65% of all life on Earth. All non-avian dinosaurs perish, marking the end of their reign as a dominant terrestrial vertebrates. The Kt extinction occurred approximately 66 million years ago. It was likely caused by a combination of factors including an asteroid impact and volcanic activity, resulted in the extinction of approximately 65% of all plant and animal species. It led to the demise of non-avian dinosaurs, paving the way for the rise of mammals. Moving on to the Cenozoic era, the Paleocene was when the angiosperms of flowering plants experienced expansion and diversification. Forests undergo rapid growth in the absence of large herbivorous dinosaurs. Mammals emerged as dominant terrestrial vertebrates. Mammals, initially small in size, rapidly evolved to occupy larger ecological niches. The Eocene, marked by a very warm climate with forests extending to even the poles, ungulates or hoofed mammals rise to prominence. Whales experienced diversification and evolutionary expansion; grasses emerged in wet herbicide habitats. The epoch concludes with the OI1 event, which signifies the onset of significant cooling and expansion of the Arctic ice. The Oleocene was when Antarctica becomes covered in ice, marking a significant cooling trend. Most modern families of mammals at present, representing a diverse array of terrestrial vertebrates. Hominids, ancestors of apes and humans, make their first appearance, marking a crucial step in primate evolution. Angiosperms continue to dominate terrestrial flora, shaping ecosystems worldwide. Crassus spread beyond waterside habitats, expanding their ecological footprint. The Miocene was when grasslands emerged, leading to an increase in grazing animals. Hominids or bipedal apes, including early human ancestors, evolved and inhabit the Earth. The Earth experiences a period of warming followed by a cooling trend, shaping global climate patterns. The Pliocene was when the Australopithecus early hominids appear, representing an important stage in human evolution. The Great American Interchange occurs, enabling the exchange of species between North and South America. Panama arises from the seafloor, connecting North and South America for the first time since the Cretaceous period. The Pleistocene era was when modern humans Homo sapiens appear, marking the beginning of the Paleolithic period. Extinction of megafauna, including iconic species like the mammoth, mastodons, glyptodons, and giant ground sloths, occurs. Mammoths were large elephant-like mammals adapted to cold climates that roamed the earth. Mastodons were distant relatives of elephants that inhabit various ecosystems across North America. Glyptodons were armored mammals resembling giant armadillos that thrived in South America, while giant ground sloths were massive herbivores with unique adaptations that grazed in the landscape of the Americas. The Holocene was when the glacial retreat marks the end of the Ice Age, worldwide expansion of human population, commencement of recorded human history, marking the beginning of modern civilization. And we have a few mnemonics that will help us remember the epochs, which are the Paleocene, Eocene, Oleocene, Miocene, Pliocene, Pleistocene, and Holocene, which are party elephants only make people happy, or pretty eager old men play poker hard. Mnemonics to memorize all periods are "Come over someday, maybe play poker, three jacks can take queens", or "Charlie Oliver still drives my purple Plymouth to Jersey City through quicksand".
Common ancestry. What is common ancestry? It's the scientific belief that organisms that share traits descend from the same ancestors at different points in time. Similarities between DNA, morphological, and behavioral traits provides evidence for shared ancestry. When comparing DNA sequences, it can show how different species are related. Fossils are a key point to providing evidence of long-term evolutionary changes, as well as for documenting species that are now extinct. A key term to be known is biogeography, which is the study of geographical distribution of species, which can help construct the timeline of evolution. How do we study common ancestry? We study common ancestry using cladograms. Cladograms are also known as phylogenetic trees. They are depictions of the common ancestry. They are also evolutionary family trees. These evolutionary family trees show the relationships between living things. One well-known example of a phylogeny or a evolutionary relationship would be the evolution of primates. Parts of a cladogram. Each of these letters is a species, which is also referred to as a taxa, and it's a leaf on the tree, this phylogenetic tree. So A, B, C, D, E, these are all leaves. Each branch point, like this yellow dot here, are referred to as nodes, and it's the most recent theoretical common ancestor for two taxa, or for more taxa. Like this is for A and B, this would be for A, B, C, and D, and then this would be for the whole tree. The root, as we can see here, is the first hypothetical ancestor in the entire tree. Known species are always leaves, they're always taxa, because species ancestors and relatives cannot be told apart. The biological species concept. According to the College Board, it is organisms of the same species if and only if they can interbreed and produce viable fertile offspring. And one limitation of this biological species concept is that it requires reproductive barriers. But we'll get more into that later. Speciation. Speciation is the process of a species turning into two or more species due to evolutionary changes over time. As we can see, the Galapagos finches is a very famous example of this, multiple species of finch. How does speciation occur? Speciation can only occur when there are reproductive barriers that are causing separation, whether it's a pre-zygotic barrier, which is preventing mating from occurring, or a post-zygotic barrier, which prevents the creation of viable fertile offspring. There are two types of speciation, allopatric and sympatric speciation. Allopatric speciation is when populations become isolated from each other geographically, as we can see in this example here, the river forms between the trees, and populations are unable to interbreed, which prevents gene flow. Then new species appear in different areas. Then there's sympatric speciation, as we can see here. Populations in a habitat become reproductively isolated, and then new species appear in the same areas. The types of separation. There's vicariance and migration. Vicariance, as we can see at the top, is when the separation of a population into isolated groups is caused by geological or climate events, like the breaking off of an island, or tectonic plate movements. And then there's migration, when the movement of individuals, either anthropogenically or naturally, occurs from one area to another. Another key topic or term to know is extinctions. They were previously mentioned before, all of the major extinction events by Aeon, but here, extinction is just the death of an entire species when every single member of that species ends up dead, and it wreaks havoc on ecosystems. Or not, it can be beneficial too. We'll get more into that later. There have been multiple mass extinction events, which is when a major portion of Earth's species die off in a short period of time. We mentioned five, or the five previously major extinction events. On the bright side, a species extinction opens up roles and niches for other or new species to occupy. As you can see in this picture, this is an example of an extinction, the KT extinction, which killed all the dinosaurs. Adaptive radiation. Adaptive radiation is a form of speciation in which the rapid diversification of organisms from an ancestral species into multiple species or forms occurs. It's commonly related to divergent evolution. As we can see here with the radiation of marsupials, one ancestral species became all of these species through speciation. Or another famous example would be the Galapagos finches. Equilibrium. Starting off with gradualism, it's a proposed theory of evolutionary speciation process which takes place slowly with small changes over a long period of time. The changes add up and accumulate into different species, as we can see here. Then there's punctuated equilibrium, which proposes that species remain in stasis for large periods and then occasionally undergo rapid change, like here. And then there's the species extinction equilibrium, which is the concept that species extinction rate with the death of species and the speciation rate, the creation of new species, is constantly equal. So biological diversity remains constant and balanced within a stable ecosystem, and this helps keep ecosystems stable. Next, we'll be talking about cladistics. What is cladistics? It's the method/approach to biological classification based on a hypothesized or most recent common ancestry. Here's an example of a cladogram. Before we get into cladistics, let's talk about the Linnaean classification system, which is the hierarchy of organisms. As you probably know this famous acronym: kingdom, phylum, class, order, family, genus, species. It was the way of organizing organisms, both plants, animals, and others. It's not typically used that much anymore. And then there's binomial nomenclature, which is the scientific system for naming species. It consists of a genus name and the species epithet. It must be italicized or underlined. Here are three species: Felis catus, Velociraptor mongoliensis, and Panthera tigris, the house cat, the velociraptor, and the tiger. Taxonomy, which is the scientific method for both naming and classifying organisms. It's based on evolutionary relationships between organisms, which we determine based on their phylogenies. There are three types of taxonomic groups. A monophyletic group, which is the best, or best in the terms of cladistics. It's a type of group that includes all of the descendants of one singular ancestral species. Then there's a paraphyletic group, a group that includes some but not all of the descendants of one ancestral species. Then there's polyphyletic. It's kind of the worst group. A group of species that are related from different lineages and not necessarily descendants of only one common ancestor. Another thing to take into account for taxonomy is outgroups. Outgroups are the most distantly related taxa or species, used to separate informative and uninformative traits. We'll get into that now. When creating a phylogeny, you must have inheritable character traits that can be compared between organisms, and then those inheritable traits are then used with parsimonious analysis to combine to make cladistics. 3 types of traits we'll look at: synapomorphies, which are shared derived characteristics, and they are informative of common ancestry of organisms; plesiomorphies, which are shared ancestral traits, so they evolved first, and they are uninformative of common ancestry; and then autapomorphies, which are unique traits that are only present in a single taxon or single species. They are also uninformative. It's important to note that all phylogenies are hypotheses. There's nothing concrete about them. Now, I have to mention this, that the AP does phylogenies differently than the way Mr. Cahill taught. So the AP does it based that taxas are grouped based only on similarity, or completely on similarity. And then this example I took from Mr. Cahill's PowerPoint. Look, they are put together not based on evolutionary changes, but based on how similar they are, based on traits. Parsimony, or parsimonious analysis. It's a scientific principle that the most direct and simple solution is the most likely correct solution. In the case of cladistics, the less evolutionary changes, the more parsimonious the hypothesis. The least change is the best and most probable answer. As Mr. Cahill is fond of saying, evolution doesn't like to reinvent the wheel. And then this is an example of a parsimonious cladogram, or should be. Continued. A character matrix is a table of organization of species and shared traits. You have the traits up at the top and the species down on the bottom. The columns are traits and the rows are species. A 1 means that the trait is present, and a 0 means the trait is not there, it's absent. And this helps us determine in-groups and out-groups. In-groups are taxa that are being observed to determine their relationships, whereas out-groups are related taxa which are used as a reference to determine informative traits. In this case, angelfish would be the outgroup, and the in-group would be everything we're comparing, so the rest of them. If an outgroup has a trait, that trait is considered an ancestral trait because it evolved before the in-group, so that makes it a plesiomorphy, and that's uninformative. Outgroups are used to determine synapomorphies and then plesiomorphies, like the difference between them, whether a trait is either one. Oh no, a homoplasy. What is a homoplasy? Homoplasy are traits that are shared between organisms, but not, very important, not because of shared ancestry. Cladistics favors homologies over homoplasies. Homoplasies involve more evolutionary changes, so they're less parsimonious, which makes them less accurate, or less scientific. Tree topology. Topology is just the shape of the phylogenetic tree. So here at the top, these are all the exact same tree, like the relationship is the same, but it's different, like they're arranged differently, or it looks different. However, on the bottom, these are different trees arranged differently. A polytomy is a tree that is not fully resolved or solved. It can have more than one correct tree. And there are 2 types of polytomies. Hard polytomies, which is due to evolution, it's true, and then soft polytomy, which is a result of bad data. Taxonomic definitions. There are 3 types of taxonomic definitions. Apomorphy-based clades, which is the first ancestor, like here, to develop a new trait, and then all of his descendants. So this is an apomorphy. This node over here, and then its descendants. This is an apomorphy. Then node-based clades, where it's 2 taxas, most recent common ancestor, and then all of the descendants of that common ancestor. And then there's stem-based clades, where all organisms that are closely related to one taxa than they are to another. Taxonomic groups, or species groups, are sometimes referred to as clades. Now, getting towards the end. Character optimization. When you place traits in a phylogeny in the most optimal or most parsimonious way, this is optimization. There's 2 types, Deltran and Actran. With Deltran optimization, optimizations happen later, where they focus on convergent evolution, like we can see here. And then we have changes that typically happen later along the branches of the tree. Whereas Actran optimization, the optimizations happen faster, they happen more rapidly, and there's a focus on losses. Changes typically happen earlier on, and then they're added back or removed in between. The changes typically happen earlier on in the tree. Thank you so much for listening.